Alexander Fleming, a Scottish biologist, changed the course of medicine in 1928 with a serendipitous discovery. Fleming was known for his keen observation skills and sometimes messy laboratory practices. Upon returning from a vacation, he noticed something peculiar in his petri dishes. A mold had contaminated a culture of Staphylococcus bacteria, a common cause of infections. Intriguingly, the area around the mold was clear, indicating the bacteria had not grown there. This observation sparked Fleming's curiosity. He identified the mold as Penicillium notatum and suspected it produced a substance that inhibited bacterial growth. He named this substance penicillin. Fleming's early experiments showed that penicillin was effective against a range of bacteria. Despite its potential, Fleming faced challenges in extracting and purifying penicillin in sufficient quantities for clinical use. His initial attempts yielded limited success, and the scientific community was slow to recognize the significance of his discovery. However, Fleming remained convinced of penicillin's therapeutic potential. Fleming's early experiments with penicillin were rudimentary but groundbreaking. He found that the mold's antibacterial action was most potent when grown in specific nutrient broths. He tested penicillin on various bacterial strains, observing its effectiveness against common culprits of infections like pneumonia, meningitis, and diphtheria. He even applied penicillin directly to minor wounds and infected eyes, noting its remarkable ability to clear infections. His findings, published in 1929, initially met with skepticism. The scientific community underestimated the challenges of producing and purifying penicillin for widespread use. Undeterred, Fleming continued his research, attempting to develop methods for mass production. However, his efforts were hampered by the instability of penicillin and the difficulties in extracting it in pure and usable forms. The lack of advanced biochemical techniques at the time proved to be a significant hurdle. A decade later, the world's attention turned to penicillin as World War II raged. The need for effective treatments for battlefield infections became dire. Two scientists at Oxford University, Howard Florey, a pathologist, and Ernst Boris Chain, a biochemist, picked up the mantle. Florey and Chain, recognizing the potential of Fleming's discovery, embarked on a mission to purify and mass-produce penicillin. They assembled a team of brilliant scientists and engineers to overcome the daunting technical challenges. Chain meticulously analyzed the chemical structure of penicillin, paving the way for its synthesis. Florey, a visionary leader, spearheaded the clinical trials that would prove penicillin's effectiveness in treating bacterial infections in humans. Their combined efforts led to the first successful treatment of a human patient with penicillin in 1941. The successful development and mass production of penicillin during World War II marked a turning point in medical history. Penicillin, dubbed the miracle drug, became the first widely available antibiotic, revolutionizing the treatment of bacterial infections. Its impact on global health was immediate and profound. Penicillin saved countless lives on the battlefields and transformed civilian healthcare. Diseases that were once death sentences, such as pneumonia, syphilis, and sepsis, became treatable. The discovery of penicillin ushered in the antibiotic age, leading to the development of numerous other life-saving antibiotics. It fundamentally changed medical practice, paving the way for modern surgical procedures and organ transplants by providing a means to control infections. Penicillin's legacy continues to shape healthcare today, reminding us of the transformative power of scientific curiosity and collaboration.